Dr. David Potter, profesor asociado de Medicina de la División de Hematología, Oncología y Trasplantes, Universidad de Minnesota. Sus estudios buscan determinar si las epoxigenasas son objetivos terapéuticos contra el cáncer. Su hipótesis es que la activación de la hipoxigenasa promueve la progresión del cáncer de mama, promoviendo la fosforilación de AKT y la supervivencia de las células cancerígenas. Estos estudios promoverán un mayor desarrollo de las epoxigenasas como objetivos para la terapéutica del cáncer de mama. El Dr. Potter ha recibido diversas distinciones como el Premio Walter por su investigación en cáncer y el Premio de Enseñanza por la Universidad de Tufts. Recibamos con un gran aplauso a este gran profesor, el Dr. David Potter.
I'd like to thank the organizers of this outstanding meeting for the opportunity to present our work today, and thank Dr. Luis Herrera and the patrons of INCAN who've supported the many students who've trained in my laboratory, and I want to express our gratefulness and show you some of their work and how we want to build a better future for our patients based on our work. So the title of my talk is Heme Binding Biguanides Target the Cytochrome P450 Dependent Cancer Mitochondria. And this introductory slide summarizes the tools that we use to translate some observations that we've made to the clinic in the area of hormone therapy-resistant breast cancer. And on the left-hand side, you can see a cytochrome P450 nanodisc made by Dr. Stephen Sliger and colleagues at University of Illinois in Urbana. In the center, you can see a crystal from Dr. Tom Poulos and Irina Sevrakova at UC Irvine, where the diabetes drug metformin is crystallized in the active site of this enzyme. And this gives us a mechanistic basis of some observations that biguanide drugs may somehow be repurposed for breast cancer treatment, although what I will show you is that they aren't sufficiently potent, and that using pharmaceutical chemistry, it's possible to synthesize at the very least new chemical probes to study this mechanism. And on the right-hand side, you can see hexyl benzyl biguanide, which is our chemical probe and our lead to potentially take to the clinic, although we're still not sure that this is the right molecule to move forward. I want to disclose support for our work from Immunomet Therapeutics, which is working with the Minnesota Partnership to help us on a grant to translate hexyl benzyl biguanide to the clinic. And this slide summarizes the basic mechanism that we are working on, and the idea is that cytochrome P450 enzymes can convert arachidonic acid to epoxides, and these epoxides have many properties which are unexpected. They were first studied in the regulation of blood pressure by Dr. Jorge Capdevila at Vanderbilt, and we have now shown that they play a role in as a potentiometer, if you will, of the electron transport chain. And later in my talk, I'll show you how these same molecules can potentially play a role in the transit of the estrogen receptor to the nucleus in estrogen receptor positive breast cancer. So the highlights are that cytochrome P450-3A4, which metabolizes about half of prescription drugs, in terms of its endogenous activities, processes arachidonic acid to epoxides that promote the engraftment of breast tumors, and I'll show you data for that, that these CYP3A4 enzymes suppress autophagy and promote the mTOR pathway, and at the same time shut down AMPK activity, which has a burn the furniture role in cells, and that the epoxygenase activity regulates the electron transport chain, and that metformin inhibits cancer in part by inhibiting CYP3A4 epoxygenase activity, but it isn't the right molecule to move to the clinic because of its lack of potency, and I'll show you data about that. So when I go to clinic twice a week, I sit across from women with metastatic estrogen receptor positive breast cancer, the most common type of breast cancer. And these patients face a very uncertain future. And in the ER positive HER2 negative signature, which we have maybe 30,000 patients per year, the only progress that's been made in the last 10 years or so has been the introduction of cyclin-dependent kinase inhibitors, palbociclib, ribociclib, and abemaciclib. And while these drugs do improve survival, we're really looking at only an improvement of 30 months to 38 months, and we can do better. And so what we need is more investigation to try to narrow down these pathways of resistance and develop new strategies to approach this problem. And this is the second generation of Hanahan and Weinberg's hallmarks of cancer, and what we're going to focus on today is the deregulation of cellular energetics. So we made an observation early in the course of this work that the CYP3A4 knockdown sensitizes estrogen receptor positive HER2 negative breast cancer cells to tamoxifen. And what we also found was that the cognate products of this enzyme restore the growth inhibition by 4-hydroxytamoxifen. So this gave us a clue that this EET pathway may play a role in hormone therapy resistance. And the question was whether this pathway was related to the known pathways of resistance and whether breast cancer that's resistant can be made sensitive by taking away these EETs. So this slide shows our approach, which has been focusing on the tumor cells, on the intrinsic cytochrome P450 enzymes that are expressed not just in the liver where they metabolize drugs, but in the tumors where they can make these EET products that promote breast cancer engraftment and growth. And this slide is from one of my colleagues, Dr. Deepak Panigrahi, who has worked on the other side of this question and has looked at what happens if you use an Alzit pump to infuse large quantities of EETs, and indeed you promote escape from tumor dormancy and you promote angiogenesis. So there are many aspects to how these EETs work. And this slide shows the many pathways for processing of arachidonic acid from lipids, and this from phospholipids, and this shows the COX, LOX, and EPOX pathways. And the EPOX pathway that we study on the right is less well studied because it requires fairly detailed methods of mass spectrometry to measure the production of the EET regioisomers. And so we came to the conclusion that the epoxygenase is functioning as a supportive non-oncogene, which by itself doesn't have transforming capability, but which plays a very important supporting role. And if we can block that role, we might be able to make some progress. So CYP3A4 makes more EETs than HEATs, which are other products made by lipoxygenases. And the 14-15 regioisomer increases the breast cancer mitotic index and the clonogenicity of these cells. And on the other side of the coin from Dr. Panagrahi and colleagues, these EETs promote multi-organ metastasis and escape from tumor dormancy. So when we looked at the engraftment process, what we saw is that if you knock down CYP3A4 stably, you don't get tumors. But what you do get on the right is little nubbins of living tumor cells which are unable to form a full-fledged tumor. And so we asked the question, how does CYP3A4 affect breast cancer growth? And we came up with two hypotheses. One of them was mitochondrial mechanisms, because we'd seen some connections with mitochondrial function, and nuclear mechanisms, because those who have been working on metformin and diabetes, specifically Dr. Alex Soukas at Mass General Hospital in Boston, has shown a role for metformin in impacting nuclear transit of mTOR components. So let's focus first on the mitochondrial mechanisms. So what Dr. Shijin Guo in our laboratory showed is that CYP3A4 is present on breast cancer mitochondria. And so MitoTracker measures mitochondria. CYP3A4 antibody detects CYP3A4. And when you look at the merge, what's particularly interesting is that there is a ring of perinuclear mitochondria surrounding the nucleus. And we have found in subsequent studies that I'll show you that this localization in the cell is important not only for energetics, but also for nuclear transit of important factors. And so what we found is that if we throw in soluble epoxide hydrolases and inhibitors from Dr. Bruce Hammack's group at UC Davis, we can promote the oxygen consumption rate in tumor cells. And this was a primary effect, and less effect on ECAR, which is measurement of lactate production. So we see an increase of oxygen consumption, the OCR, and not much effect on the lactate, which is a secondary effect. And so with Dr. John Falk at UT Southwestern and John Immig at Medical College of Wisconsin, we tested one of their new analogs for EETs, and we found that this non-hydrolyzable analog, if anything, further increased oxygen consumption rates, suggesting that there was an impact on the electron transport chain. And using antagonists, 1415 EEZE, which suppresses OCAR and ECAR and activates AMPK, we see the opposite effect, suggesting that these EETs are playing a role in the oxygen consumption rate. And so we asked the question, does CYP3A4 affect AMPK? AMPK has this burn the furniture effect on cancer cells, where the cancer cells are undergoing autophagy to try to survive under conditions of stress. And what we found is that CYP3A4 knockdown activates AMPK similar to metformin, and that the EET products of CYP3A4 inhibit AMPK, suggesting that the EETs were playing some central role as a second messenger. And so we looked at work that was done by other groups, and Dr. Michael Pollack showed in 2006 that metformin is an AMP kinase-dependent growth inhibitor for breast cancer cells. And we showed that CYP3A4 makes EETs that activate the STAT3 pathway. Bringing these two observations together, Ann Thor's group at University of Colorado showed that metformin targets STAT3 to inhibit breast cancer cell growth and induce apoptosis. So we then asked the question about whether metformin could substitute for the CYP3A4 knockdown, because metformin activates AMPK. So the question was, does metformin inhibit CYP3A4 in tissue culture and in tumors? And if metformin inhibits CYP3A4, what are those metabolic products? And to attack this problem, we realized early on that metformin just isn't very potent. And so what we needed to do was develop new chemical probes such as hexyl benzyl biguanide. And although hexyl benzyl biguanide may or may not be the right drug to develop for the clinic, what we needed to do was to have a highly potent and specific derivative of metformin that primarily attacked the target that we were interested in. In contrast, drugs need to be safe and effective, and so they do not need to be as specific. So we asked, first of all, what does metformin do to EET biosynthesis by mass spectrometry? And Dr. Guo found that as we titrate up metformin through the millimolar range, we get inhibition of the production of these EET products. And if you use microsomes, you see the same effect. The crystal structure showed us the coordinates of metformin bound in the active site of CYP3A4, hovering over the biguanide and hydrogen bonded to arginine 212. This work was later confirmed by Dr. Michael Snyder's group at Stanford, where they found that metformin interacts with many different heme-containing proteins. And one of the ideas is that the conjugated pi system of metformin can hover over the conjugated pi system of the heme ring. And so, working with Dr. Steven Sliger and Ilya Denisov, we took these CYP3A4 nanodiscs and synthesized a number of different compounds that were derivatives of metformin but had more hydrophobic arms. And these compounds were binding by Dock score with much, much higher affinity than metformin to the CYP3A4 metformin co-crystal. And those Dock scores were at least 3 orders of magnitude higher. And this work was done by Dr. Elizabeth Ambrose in our College of Pharmacy. So our College of Pharmacy, through Dr. Gunda Georg, synthesized the HBB and a number of different analogs. HBB appeared to be the most straightforward to synthesize and was a useful chemical probe that inhibited the target. And HBB was 200-fold more potent than metformin and inhibits the necessary target. But at the same time, we sent this molecule to Dr. Martin Brand's group at the Buck Institute in Novato, California, and they tested whether it inhibited mitochondrial glycerol phosphate dehydrogenase, which plays a major role in gluconeogenesis and is thought to be a major target of metformin from the work of Madaraju et al. At Yale and Jerry Schulman's group. And if you think of the mitochondrion as being this wire, if you will, with a number of appliances attached to the wire that feed off of it, mitochondrial glycerol phosphate dehydrogenase is involved in shuttling electrons and plays a major role in gluconeogenesis. And indeed, HBB inhibited this very same target with 200-fold greater potency than metformin. So we thought we were on the right track. And we also had a modicum of specificity because when we looked at other CYP enzymes, which are heme-containing, we saw that 3A4 had an IC50 of 9 micromolar as opposed to CYP2C8, which was 50 micromolar, and we looked at a host of other CYP enzymes that were even less inhibited. So there was some specificity between those sister CYPs, and which all contain heme. When we looked at whether the EET or the C22 analog could restore growth, we saw for metformin, buformin, fenformin, and HBB, we could restore growth. But as we got more potent, the analogs were less and less effective at restoring growth, and there was no activity in restoring the growth of triple-negative breast cancer, suggesting that we were on the right track to be looking at ER-positive, HER2-negative breast cancer. We then went back to metformin and to the seahorse machine, and we measured how well metformin inhibited OCAR and ECAR, and it really had very little effect, and there was little effect on ATP production, even in the millimolar range. But at the 20 micromolar range, HBB was able to potently inhibit OCAR and ECAR, the oxygen consumption and lactate production, and reduce ATP. And this was reproducible in a variety of breast cancer cell lines. And we found that HBB inhibited STAT3, later activates AMPK, and inhibits mTOR. So it had all of the features of what we would expect. And when we went to a mouse model, we found that HBB inhibits MCF7 tumor growth, the ER-positive, HER2-negative breast cancer cell line. But what we found is that there was some reduction of weight, and there was a limitation on the maximum dose that we could give to the mice. And there was a 50% reduction in tumor weight. But this still didn't look quite good enough to move forward to the clinic. And so what we're doing is we're developing what we think are better analogs, and hopefully in the future I'll be able to tell you more about those new analogs. What we did find in the tumor model by reverse phase array analysis with Chip Petricoin and Julia Wolfkuhl, that mTOR levels were reduced, not the phosphorylation, but the levels, and that PKC lambda zeta, which is important in insulin signaling, was reduced. And so we have CYP enzymes in different locations in the cell, and we were interested in whether exogenous or membrane EETs actually reach the mitochondria, or is it important that they're made on-site close to the mitochondria and potentially close to the nucleus, where I'll show you other effects may be had. And that we wonder also whether these biguanides inhibit different functions of CYPs in different locations. So we have drugs that are perhaps a little less specific, but again, we keep our eye on the clinic, because if we can move them forward and they're safe and effective, but adequately potent for the cancer problem, we're hoping that we can make progress in hormone therapy resistant ER-positive, HER2-negative breast cancer. So now I'm going to tell you a little bit about the nuclear mechanisms. So we had shown in a JBC article in 2011 that EETs promote the nuclear transit of phospho-STAT3. And Alex Soukas gave a really wonderful talk at a Cold Spring Harbor meeting, which I was lucky enough to attend about a year and a half ago. And what he showed is that metformin inhibits the growth of C. elegans, but improves their lifespan. And what he found is that under normal conditions of growth, there is a component of the mTOR complex called RAGC that David Sabatini had first identified. And this RAGC has GTP on it, goes into the nucleus, gets hydrolyzed to GDP, and then RAGC with GDP comes out of the nucleus and then activates the mTORC1 complex. And when that happens, an important effect of mTOR is that it suppresses the breakdown of biomass, and specifically what's inhibited is an NRF2 to ACAD10 pathway. So ACAD10 is an acyl-CoA dehydrogenase that's involved in the oxidation, beta oxidation of lipids. And when you shut down this beta oxidation of lipids, it promotes lipid retention and helps promote biomass assembly. So it suggested that metformin played some role in the nuclear pore complex, but what was not known was what was the second messenger and where was this signal coming from that could regulate the nuclear transit of RAGC. And so based on the prior data, we asked whether HBB could impact the nuclear pore complex. So using digitonin permeabilized cells, we found that HBB inhibited nuclear localization of FTSE 70 kilodalton dextran. So we asked whether RAGC nuclear localization was inhibited by the genetics. So we looked at CYP3A4 knockdown in MCF7 cells, and we found that RAGC levels in the nucleus were reduced in a steady state way. So then we asked whether we could use HBB as a chemical probe to perturb the system over short time periods, and we found that as we went 1 hour, then 6 hours, we could reduce nuclear RAGC levels. And nuclear RAGC was also decreased in T47D, another cell line, and we found that there was a correlation between cytoplasmic CYP3A4 and nuclear estrogen receptor alpha in some beautiful immunofluorescence studies that were done in collaboration by Dr. Daffet Thomas at University of Michigan. And so then we asked, what about ER alpha? And we found that nuclear ER alpha is decreased in MCF7 cells treated with HBB at 2 hours. And this was true in T47D cells, which are also estrogen receptor positive and HER2 negative, and also in the ZR75 cell line. So this wasn't an effect that was specific to an individual cell line. And when we did qPCR, we found that decreased estrogen receptor alpha induced by the HBB drug resulted in reduction of 3 critical genes expressions that are tied to breast cancer progression. One of them being the estrogen receptor, ESR1, which drives its own transcription, the progesterone receptor, and SLC7A5, which correlates with very poor prognosis in luminal B breast cancer and is a leucine transporter that helps promote this anabolic tumor cell growth pathway. And so we then asked, if you just took the EETs, could that force estrogen receptor alpha into the nucleus? And so Dr. Jianxun Li, who is a new postdoc in the lab, tested this in the past couple of months. And what he found is that in MCF7 cells and ZR75 cells, which are ER positive, HER2 negative, he could actually force estrogen receptor into the nucleus, suggesting that these EETs may play an important role in the function of the nuclear estrogen receptor in breast cancer and potentially might play a role in resistance to hormonal therapy. And so we now have a new arm of the EET pathway. So I showed you that those mitochondria are located close to the nucleus, containing some piggybacked CYP3A4, and that there was an effect on the electron transport chain. Now we're seeing that there's also an effect on the nuclear localization of the estrogen receptor. And we performed RNA-seq analysis to look at the whole transcriptome, and what we found is that there are potential resistance pathways that have been activated, including the RAS pathway and also the IGF pathway, which is studied by Dr. Doug Yee at the University of Minnesota. And so this slide summarizes the overarching mechanisms that we're working on, that the CYP associated with perinuclear mitochondria has some impact on the electron transport chain, and that these same EETs can play a role as a second messenger impacting the nuclear pore complex, allowing RAGC to transit through the nucleus and drive mTOR functioning, and that ER alpha and STAT3 also can be forced into the nucleus to drive a transcriptional program. So the possible implications are that targeting the ETs may have an impact on the function of ER alpha and STAT3 and the function of RAG-C. So these are things that we have yet to demonstrate. But it also suggests that an explanation for the synergy that was found by Dr. Jose Baselga when he conducted a clinical trial looking at everolimus, which is an mTOR inhibitor, in combination with exemestane, which is an aromatase inhibitor, and the combination exhibited synergy. These studies may provide some mechanism for why that combination may be effective. So here's what we are facing in the clinic, the challenge that we have every week as we go to clinic. And we see patients with hormone therapy resistant metastatic breast cancer. And we have aromatase inhibitors. Now we have cyclin-dependent kinase inhibitors. And we have mTOR inhibitors that don't really impact survival. We now have PROTACs that are coming to the clinic. So there's a new generation of agents that interact with E3 ubiquitin ligases and help degrade the estrogen receptor. Fulvestrant was kind of the prototype. And there also is the potential for immunotherapies to move forward to the clinic as well, as we learned from the I-SPY 2 neoadjuvant study. Whether those will work in the metastatic setting remains to be determined. And so far, we don't know how to do it right. And so early work from Hope Rugo and colleagues really doesn't show much promise for immunotherapy in ER-positive, HER2-negative. But we might be able to do better by improving the function of T cells in that context. And that's other work that we're working on, but I'm not going to talk about today. So what we wanted to do in terms of our development grant to bring HBB or an HBB-like molecule to the clinic is we wanted to know if we looked at models of hormone therapy resistance, was there resistance also to the HBB chemical probe? And what we found in Angela Brody's aromatase inhibitor expressing cells, we don't see any resistance. We also looked at Peter Schmidt's ribociclib and palbociclib resistant cells, and they don't show any resistance with IC50s in the couple micromolar range. And then finally, 30% of hormone therapy resistant breast cancer has mutation in the hormone binding domain of ESR1, which constitutively activates the ER alpha. And when we look at a CRISPR knock-in of these mutants in MCF7, none of these cell lines are resistant to HBB, and they're all sensitive in the 10 micromolar range for IC50. And this is cell lines that we're collaborating on with Steffi Ostrich at University of Pittsburgh and Dr. Ben-Ho Park at Vanderbilt. So in summary, CYP3A4 is an arachidonic acid epoxygenase that's required for breast tumor formation, that these epoxygenases play an important role in the electron transport chain, and that metformin inhibits breast cancer in part by inhibiting a structural target, a heme protein CYP3A4, and its cognate arachidonic acid epoxygenase activity, and the eicosanoids may link ER alpha and mTOR function in breast cancer. So I want to thank INCAN for your very kind, and the patrons for your very kind support of the many students that we've been privileged enough to have in our laboratory. And I must say that these are outstanding students who are the best of the best. And it's just been an outstanding privilege to have them in our laboratory. Our work is also supported by our Masonic Cancer Center, Dr. Doug Yee from our, and Dr. Chuck Ryan from our Division of Hematology Oncology, a grant from the Department of Defense to develop immunotherapy with HBB, from the St. Paul Foundation, which has funded work to build new biguanides, and INCAN, of course. And this work started with with early support from, it was an outgrowth of an NIH grant and grants from Susan G. Komen and Flight Attendants Medical Research Institute, and other support as noted, including a brainstorm grant from our Cancer Center to support immunotherapy approaches, and the Randy Shaver Cancer Research and Community Fund, which has supported our work from the get-go. And I want to acknowledge the people who did this work, Dr. Shijun Guo and now Dr. Jiangxun Li, who's come to the lab, many graduate students and medical students at the University of Minnesota, and for our pre-MSTP program, our colleagues that I mentioned from other institutions, and the students from INCAN mentioned here, Dr. Matt Getz at the Mayo Clinic, who's worked, and Dr. Peter Schmidt, and Steffi Ostrich and Ben Ho Park, who worked with us on the hormone therapy resistant models. And there are some pictures of everyone, and you can see Erwin Hernandez, who was our first student, and outstanding other students as well in the group photo, the cover of our cell chemical biology issue with our paper, and Goldie the gopher, who's the cutest mascot of the Big Ten. And thank you.